I want to welcome a lot of familiar faces, friends of long standing, and some new folks who are, I think, being introduced to things at Gladstone for the first time. We've set a sort of central premise for the evening, afternoon and evening, that goes something like this. Not far from here, and not too long ago, a miracle happened. And it happened right down the road in Silicon Valley. The combination of material science and engineering and mathematics and marketing and design and finance produced a set of products that have changed the world, permeated the lives of almost everyone on the planet, creating immense wealth for a few and enriching the lives of all of us. We have today with us some folks who had a hand in creating that miracle, and they'll be on the panel up here a bit later. But the premise for today is, what about those of us in life sciences? We want to do the same thing, of course, and I'd have to say we haven't quite achieved it yet. Yeah, a lot of good stuff has happened. We can cure some people with cancer, we can prevent some people's heart attacks, but we haven't yet created things that permeate into the daily lives of people and change them for the better, the way our friends down the road have done. And what might we learn from that? So we're going to borrow a page from information technology nomenclature, and for the first part of this experience today, we're going to talk about next generation technologies for life sciences. And I'm going to ask my friend and colleague Deepak Srivastava to lead that part of the program.
Well, welcome everybody. Delighted to see a lot of familiar faces in the room. As Sandy said, the real challenge for us is, can we harness new technologies to drive forward our ability to alter the course of human disease in ways that we haven't been able to in the past as we go forward. And fortunately, there have been a number of breakthroughs in the last few years, really technological breakthroughs, that are beginning to allow us to do things we couldn't even imagine doing a few years ago. And that's what we're going to focus on in the next period. I'm going to spend just a couple of minutes introducing to you some concepts about what we call cellular reprogramming technology. And really all that is that we now know enough about a cell to be able to almost at will control its fate and its behavior. And we really couldn't do this a few years ago, but we can do it now. And so I'm just going to show you a couple of movies that'll lay the foundation for a lot of what you'll hear in the subsequent snippets. And then I'm going to turn the program over to a couple of our scientists to describe some of their stories. And I'll come back up and tell you a direct story from my laboratory at the end of that. So I want to describe what you see here on the slide, and then I'll play a movie for you that'll make it a little more clear. This is imagine a hill where these balls are placed at the top of the hill, and they have the option to roll down this hill and go into any one of the valleys that you see towards the bottom. The valleys represent specific cell type, let's say a heart cell or a brain cell or a liver cell, and the ball at the top represents a stem cell that still has the potential to go into any one of those valleys. And a human embryonic stem cell has the potential to turn into any of the 200 or more cell types in your body. Okay? And so what we know is that those balls at the top have the potential to roll down this hill, turn into specific cell types. What we've always thought is that once a ball occupied a valley, it was fixed there and could never go back. And a discovery by Shinya Yamanaka at our place showed that if we put 4 genes into one of those balls, it can push that ball back up the hill, so it's again repositioned to come down and become any of the cell types in the body or go down into any of those valleys. So Shinya termed those induced pluripotent stem cells, because pluripotent means it has the potential to turn into any cell type in the body, and it's a stem cell. And we've induced that by putting in a group of just 4 genes into that cell. Now, that discovery changed the way we thought about cells in the body, that we could even push these cells back up the hill and change their fate. It, as many of you know, received the Nobel Prize in 2012 because it so fundamentally altered the way we thought about cells. Now, you may ask, once we put a ball back up the hill and it's poised again, we don't want it to randomly go into any valley, but let's say we wanted to make a brain cell or a heart cell, we have to tell it to do that. And so over the last several years, we've had many breakthroughs that allow us now to very efficiently tell the ball to go into one valley and not all the others, and we can get pure, relatively pure cultures of many different cell types. And schematically, that you can conceptualize something like this, where we, whoops, I should forward this slide, where we essentially erect barriers to certain valleys, and we leave one valley open. And in doing so, we essentially now let the cell, guide the cell into one specific cell type, and we don't let it go anywhere else. And this is what we can do now for heart cells, brain cells, pancreatic cells, liver cells, et cetera. Now, what we've been able to do most recently is sort of take this to the next generation, if you will, of cellular reprogramming technology, what I like to call reprogramming 2.0, again, to take a riff off the technology community here. And here the notion is, what if we wanted to make a heart cell, let's say, from a skin cell, or a brain cell from a skin cell? We can push the ball back up the hill and then direct it back down. What if we could be more efficient and just take a skin cell and turn it directly into a heart cell or a brain cell without becoming a stem cell? And that would look something like this, where we take a cell that's already in one of these valleys and somehow add the right cocktail of factors that make them not go back up the hill, but rather jump over and go directly into a different valley. So in the last 2 years, we've been able to do that for heart cells, brain cells, liver cells, and pancreatic insulin-producing cells, as well as several other cell types. So we think now we can actually do this, and this opens up whole new possibilities for how we could harness this technology in the coming years. So I just wanted to show you those movies so you have a conceptual framework for what we mean by cellular reprogramming as you hear the next few talks, which will describe specific uses now for this technology to address human diseases. So with that, I'm going to introduce our next speaker, Dr. Kathryn Ivey, who directs our stem cell core here and is one of our young scientists.
Hi. So now you sort of have an idea, a conceptual idea of the idea of reprogramming, and I want to talk with you a little bit about some of the practical aspects, especially in regard to the reprogramming of adult cells into induced pluripotent stem cells, or iPS cells. So the description of this, which came out from Shinya Yamanaka in 2007, was definitely remarkable and game-changing. But that first generation of protocols that we were using to make iPS cells, I would equate loosely to rubbing two sticks together to start a fire. They're remarkably inefficient and time-consuming, and they required patience and some skill and finesse from the scientists that were undertaking that. Not that they weren't sophisticated, but they were difficult. And another difficulty was that some of the qualities of those cells were undesirable when we thought about using them for future clinical applications. One of the problems was that we were using a method that introduced genetic material right into the genes of those adult cells. And so that left a genetic footprint, and we wouldn't want to use having a genetic footprint in cells that we use for laboratory purposes, for research purposes is fine, but we wouldn't want to think about reintroducing cells with an outside genetic footprint back into people for regenerative medicine purposes. So now the second generation of iPS derivation protocols that we're using involve the transient introduction of reprogramming factors, so that there's no genetic footprint. There's no actual change to the genetic material of those adult cells as they are reprogrammed to induce pluripotent stem cells. And so this has created a fantastic platform for what we call personalized medicine, because we can use these iPS cells to derive all of the 200 or so types of cells in the body, and then start to use them as a platform to both study disease and then look for drug treatments that could counteract what we see in that disease. And we call it personalized medicine because we can also think about, we can actually test different drugs and how they would affect a particular cell given the genetic makeup of the person from whom they were derived. And now that we're also able to make clinical grade iPS cells, we can start to use these, and there's promise for them in regenerative medicine applications. And in the next 6 to 12 months, this will actually be put into studies in clinical trials, specifically for applications of retinal regeneration. So I'll turn it over now to Yadong Huang, who will tell you more about applications specifically for neurological disease.
So first, I would like also welcome everyone here. So as Casey said, the research in my laboratory has been focusing on this aging-related degenerative disease like Alzheimer's disease. Probably many of you will wonder why we are interested in stem cell if we are working on these aging-related diseases. However, there are many reasons. So let me just give you 2 reasons why we are interested in stem cell work in the degenerative disease. First, now we can take the tissue biopsies from skin, from even liver, some other tissue, but it would be very, very hard to ask anybody to give this brain biopsy to study the human neurons in culture. So that gives us, if you imagine this picture, so if we can, reprogram the human skin cells back all the way to pluripotent stem cell, now we can culture the human stem cells in dish to generate human neurons and then study this aging-related disease. Let me give you one of the examples we are working on. Have been working a long time with Bob Melisade here on one particular protein called apolipoprotein E4. There are 3 isoforms in humans. E3 is the normal form. E4 is the one related to Alzheimer's disease. And then it's hard to study in human neurons. Now with this technology, we can take the skin cells from E4 carriers and also from E3 carriers, turn them back to the iPS cells, and then make again to the brain cells. Now we can study this E4 effect in human neurons in culture. By doing that, we notice this E4, because this mutation, one particular position mutation, that makes E4, the structure is very close. If you look at the E3 normal structure, they are open. The E3 like this in general, but E4 is closed. So because this unique structure making E4 more susceptible to a protease we call that cut this E4 generate toxic fragment. Now by using the human neurons in culture, we prove we do see this fragment in the culture. Now get to the second reason why we wanted these stem cells, because now we have human neuron model we can screen for drugs. So based on this unique E4 closed structure, we wonder whether we can test any compound that has a capability to open the E4 structure, making it more like E3. So by screening many compounds, we did get some positive hit. And then now we test in these human neurons in the presence of E4. And the good news here is we do see some compound when you add to the human neuron culture, they do open the E4 structure more like E3, and then we will see they decrease the fragment, toxic fragment levels, and then they protect these human neurons. So of course, the next step will be to test this compound in human trials. So now let's go to another example. That is, Deepak mentioned, now not only we can all the way push this skin cell back, all the way back to the stem cell, but we can turn them directly into other cell lineage, like we can make skin cell directly to brain cells, which will be much faster and easier. And then for the past 2 years, we have developed a protocol in my laboratory that just use one factor we call SOX2 plus very unique culture conditions, we can turn actually the human skin cells directly to brain stem cell we call neural stem cell, which is different from the pluripotent stem cell, which generate any type of cell in our body. But this neural stem cell only generates brain cells, basically 3 types of cells: neurons, glial cells, and also oligodendrocyte. By doing that, now we have a way directly convert human skin cells to brain cells. And then after we published this work, actually in the last 2 years, there's another laboratory following our work. They can do even more efficiently in vivo. They can put this SOX2 directly into the mouse brains and then turn some astrocytes, those supporting cells, into neurons in vivo. So basically, Deepak called this secondary, reprogram 2.0. We think that will be very, very useful in the future, especially for stem cell therapy in those aging-related diseases. Thank you so much.
Thanks, Yadong. So what you heard from Yadong was this notion that we could now use human cells in a dish to try to discover new drugs that could get into people ultimately, because that might be a better model than testing on an animal cell. And in fact, some of you may have heard yesterday, Bristol Myers Squibb acquired a company called Hyperion, started here in South San Francisco, that developed the first therapeutic that will likely end up in clinical trials using iPS cells as a platform for discovery. And Hyperion, in fact, was started right here in the Gladstone building as a startup company 7 years ago now, right as this technology was emerging. And so it's a wonderful story of right here in the Bay Area. The technology started right here, and the first therapeutic application of it likely will arise from right here. So Yadong mentioned this notion of in vivo converting a cell. So let me elaborate on that a bit, because this is something that my laboratory described for the first time a few years ago, and now I think you'll see this paradigm will likely apply to many, many tissue injury in the body. So you all have heard about the notion of taking a stem cell in a dish, let's say maybe even a heart cell, and then injecting that into the heart to try to create new heart cells, because after you have a heart attack, the heart can't regenerate itself and you're left with a damaged heart. That leads to heart failure, and there are 23 million people in the world with heart failure, and there are very few heart transplants done that are available. And so the 2-year mortality for end-stage heart failure is actually 50%, and we don't have any treatment for it. So it turns out that the human adult heart, every heart in this room, is only half muscle. And the other half of your heart are these support cells that we call fibroblasts that are just sitting there, never meant to be muscle, but they're in the organ. So we asked, if we can control cell fate at will, like I told you, then maybe we could take those fibroblasts that are already in your heart, after you've had a heart attack and you have a big dead muscle area, and what if we could convert those fibroblasts already in your heart into brand new muscle without ever having to put any cells in? That would be fantastic, because it's your own cells. They're already there. We don't have to put them in and get them to survive and integrate and all those things that would have to otherwise happen. And so we've been able to do that in the last few years. So far in animals, we've found that a cocktail of just 3 factors are powerful enough to reprogram the non-muscle cell in your heart directly into a muscle cell. And that muscle cell not only turns into muscle, it beats, it integrates with its neighbors, because that's critical. It has to beat in unison. The heart beats all at the same time. So they electrically connect. And in mice, we've shown that when you put these little mice in an MRI machine, which we have for little mice, we sedate them like we do people, that their hearts beat with greater force and can pump more blood after we've injured them, mimicking a heart attack, if we can reprogram their cells. So that's been very encouraging to us in the field. And we've now moved that into pigs, because their heart is bigger and more similar to our hearts and in size and physiology. And so far now it looks like, and we're actually working with Al Chin, who's one of our friends here at Gladstone, is helping us with some of these studies, and there it looks like we're getting quite good reprogramming in vivo in the pig heart with this strategy. And now we're doing MRIs on the pigs to see if we can really make a difference. And if that works, then we would move this further into clinical trials. We think this approach is not only going to be effective for the heart, but almost every tissue in the body has these support cells nearby that could be called upon if we knew enough about those cells to change their fate and reprogram them. And so I think you'll hear a lot more about this going forward, but this is a brand new technology. It's moving fast, and again, it represents one of those sort of next generation things that we couldn't even have dreamt about doing a few years ago. So we're really excited about that, and we wanted to share that advance with you guys here today. Now, next you're going to hear from Warner Greene, who's the director of our Virology and Immunology Institute, who will tell you about a different way to utilize reprogramming for a different disease.
Thank you, Deepak. I'd like to add my welcome to you this afternoon. Thank you for joining us this afternoon. It's a glorious day outside, and we're inside. I'd like to take you back over 20 years ago when I first came to Gladstone. We were at the San Francisco General Hospital, ground zero for the HIV/AIDS epidemic in San Francisco. And I remember attending on the wards my first time there. Every single patient I had for the entire month was HIV infected, and there wasn't a thing that we could do. Many of them died. All of them died ultimately. But then things changed in the mid-1990s. That was the advent of combination antiretroviral therapy. We now have 30 different antiretroviral medicines that can control this virus, but cannot cure people with this virus. The feeling was the AIDS crisis was over. You know, we had the medicines. You know, this was, we had converted an acute cancer-like condition into a chronic manageable disease like diabetes. Nothing really could be further from the truth. Today, the epidemic continues to expand. It is hitting the hardest in the developing world, where 70% of new infections occur. There have been over 70 million people infected with this virus, and over 36 million have died. Ranking it as perhaps the fourth worst epidemic behind the plague, smallpox, and the great influenza. Fourth worst epidemic in human history. So as I mentioned, even here in the United States, where we have resources and can provide medicines to all of our citizens, it might surprise you to know that only 1 in 4 individuals who are diagnosed with HIV infection are actually on effective treatment. At the end of the day, we're only really treating 25% of the population that we know is infected. Back to Africa. What's the cost of treating all those 35.3 million people in the world with antiretroviral medicines? That's what they're going to need. That is the standard of care. It's estimated that that will cost over $1 trillion in the next 20 years. Certainly a price tag that I think the developed world is going to have a hard time writing checks for. So in many ways, Africa's best hope is a cure for HIV. It was not that many years ago that we wouldn't even say the C word. The C word was off limits when it came to HIV. But things changed with a man by the name of Timothy Brown, who had HIV, but he also had a leukemia. He was transplanted twice with a particular type of bone marrow that was resistant to HIV infection. This is the single person in the world who has had the HIV disease eradicated. No trace of the virus in his body. So we've cured with an N of 1. It's possible, but that really is a game changer. It says it's not like tilting at windmills. It's a technical problem that we have to overcome. The Mississippi baby, which you've heard about, probably early treatment can nip this virus in the bud and achieve a cure. Same with the Visconti cohort. So the whole cure agenda has just had a tremendous shot in the arm. And now the search is on for a broad, effective, scalable, safe, effective, and scalable cure for HIV infection. Certainly, it's not we would not succeed if we have a treatment that might be available to a few hundred people in the richest part of the world. What we need is a scalable cure therapy that we can deploy throughout all of Africa. So, how to create a safe and effective cure? It's not easy, but right now, latent infection. So there's this thing called the latent virus, and that's what's preventing us from getting a cure with antiretroviral medicine. So I could take patients and have had patients where they have been absolutely perfect antiretroviral therapy, never had a detectable viral blip, suppressed for years. If you take those individuals off their antiretroviral medicine, the virus bounces back within weeks to the levels that existed before the infection. What's rekindling this infection in these individuals? It's the latent reservoir, a small population of memory CD4 T cells, 1 in a million. So think about this auditorium. Think about 10,000 of these auditoriums, and think about finding one person in those 10,000 auditoriums. That's what latency is about. 1 in a million cells are latently infected. And then, but the body might only contain 100,000 of those cells. But we have to be able to attack each and every one of those and eliminate them to achieve a cure. Right now, we don't know enough about the biology. We can't study those 1 in a million cells. The background is too big, so we don't really know what's going on. We have these surrogate cell models, many of which have been created here at Gladstone, but they're imperfect at best. So our idea, and it's an idea, is to use IPS technology to revolutionize HIV cure research. And the concept is to find that 1 in a million cells and convert it into an IPS cell. Now we can grow these IPS cells essentially at limitless quantities, and we can isolate clones. We can isolate pure, latently infected IPS cells. And then the concept is not to take them down another path, but exactly right back to the path that they were on. Redifferentiate them back into the memory CD4 T cell where they started out. But now it's a homogeneous population of latently infected cells that mimics the biology of the latently infected cell found 1 in a million in HIV-infected subjects. Well, what does this do for us? Well, it opens up an incredible array of studies. Now we can study what's the biological basis of latency. How is latency actually created? Is it any one of a number of mechanisms could be at play here? Well, now we're going to be able to figure it out. It also allows us to begin testing combinations of agents to try and induce that virus, to try and rally the virus out of its transcriptional slumber. This is what's called shock and kill. So the idea is to shock the latently infected cell and then kill it, and thereby trying to get rid of these few 100,000 cells, latently infected cells in the body. We know that not every cell that we shock gets killed. And so there may be the need to create new killing mechanisms to actually attack those cells. We could also look for, is there a marker on these latently infected cells? Is there some vestige of the virus actually setting up a latent infection there? That could revolutionize. If we had a cell marker for latency, it would be an incredible tool at our hand. So all of this, I'm trying to emphasize how a discovery, and I've got to get Deepak to have a slot that has immune cell. Yeah, we need an immune cell there. But how a discovery in cell biology, in development, how this IPS technology, how we may be able to apply this technology in a very meaningful way to hopefully one day achieve a cure for HIV infection. Thanks very much. It's really a pleasure for me to introduce the next speaker, Dr. Bruce Conklin. from the Gladstone Institute of Cardiovascular Disease. Bruce.
We'll definitely arrange for a different movie with the immune cells soon. I'm Bruce Conklin, and I am here to talk about the editing and controlling the human genome. Just as a way of background, I'm a physician by training, but really with my entire career, research career, has been here at Gladstone, and it's been just a tremendous privilege to really focus on different aspects of biomedicine, but particularly in the recent years, because of the discovery of iPS cells, I've been able to really start focusing on human genetics in particular, and because a lot of human genetics is actually most manifest in children, I've become more and more interested actually in pediatric disease because of that's when it's manifest in its sort of most extreme case. And when I talk about editing the human genome, I was trying to think about how I would think about what that is. And to start off, we have to start thinking about what the human genome is and how big it is. It's 3 billion base pairs is what it is, and it's A's, C's, T's, and G's. And so I tried to think of what would be a good analogy for that. And so how many people here have read Harry Potter? Okay, a good number. Okay. It's a sizable book, actually. It reads fast, but it's the first year at Hogwarts was 320 pages. So I counted the number of characters in there, or estimated, and so imagine that you had Harry Potter in the paperback form, and you have the 3 billion base pairs. How high do you think it would be? Do you think it would be, who would vote for this high for the 3 billion base pairs? Anyone? Up to the ceiling? Anyone? Okay. It goes higher than this building. So this building is 6 floors. The building's 120 feet. It would be 170 feet tall. I tried to order that from Amazon and I couldn't find it. So we'll have to just imagine a book which is 170 feet tall. Now, the problem that we have as human geneticists is that a single letter changed in that 170-foot tall pile, changed from an A to a G or a T to a C, can make the difference between a completely healthy life and someone who has cystic fibrosis, or someone who has cardiomyopathy or muscular dystrophy and spends their life in a wheelchair or dies at a very early age. A single change, a single base pair. This is actually what Sergey Brin, who carries the LARC2 mutation, calls a bug in his code. Okay, a single letter in that huge long pile can make the difference between the healthy lifestyle that many of us are very fortunate to have and tragic situations. Now, the knowledge of this is for the obvious mutations is certainly tremendous, but the ability to actually study that in people is very, very difficult because each of us is different in many different ways. Many of you have siblings, and so if we took an iPS cell from you and your brother or sister and stuff like that and compared them and tried to compare the differences, you can imagine in your 3 billion base pairs and your brothers and sisters, there's lots of other changes other than that one thing that we're trying to study. And so one of the things that we would like to do is take a page from that big pile, change one letter, and then put it back in, and then see how it runs. And to see how that change from an A to a C, for instance, in the middle of that pile will now make those cells, heart cells, beat differently, how they make neurons differently, etc. And so the task seems absolutely ridiculous. And 10 years ago, it was not ridiculous, it was impossible, right? Because we didn't even have iPS cells to make the tissues that we wanted to make, and we were just beginning to learn how to read the genome. And we've been very good at learning how to read the genome. We can actually now sequence a genome in a day, and it costs $1,000 and so on. But now editing the genome and making changes so that we can really see if that change is really the causative one is a different task. And what's remarkable is in just the last few years, we've been able to now do this so that it's possible. 4 years ago, it was a Herculean task to make a change in a human genome in iPS cells. And then just recently, in this year, my lab in particular, but and working with labs around the world, we've been able to actually make these routinely so that we can, a single postdoctoral fellow working with a technician could make multiple changes in the genome and pick and choose different places in the genome and change one letter at a time. And then we can make those cells, and we can grow them into the heart cells, we can grow them into neurons, and then we can see how they work differently. And you can imagine how powerful this is to be actually not to just be observers in the biology and just to say, well, these cells look different than those cells, but to say, this change in page, 4 stories up from here, that one little change was the one that actually caused that disease. This is something that we're applying particularly to cardiovascular disease at this point, and we're particularly interested in the pediatric diseases because they cause the largest changes and because we can see the largest phenotypes, and we are seeing phenotypes. It's remarkable. There's every day actually when we come into the lab, somebody has found something new. They say, I can't believe we just changed one residue and there's a change in the cells. The cells are behaving differently as a result of it. Even though theoretically we can talk about it, seeing is believing. But later on, as we get better and better at this, I think I believe that we can get at the more subtle changes, the types of changes which actually make somebody predisposed to adult diseases that occur later in life, the types of diseases that are much more subtle, the things that only, for instance, give you a 3-fold increase of having what we call sudden death, sudden cardiac death, or a 5-fold increase in having Parkinson's disease. Those are more subtle changes, but they're very important if you're the person who's had that heart attack or has Parkinson's. And those are the changes I think that we'll be able to get at because we're now able to make those edits and see how they go. And there's one more thing. Of course, as we get better at this, that not only are we going to be able to make these changes and edit the changes so that we can test hypotheses now and actually make disease models to now test drugs. Now we can actually really test drugs in different backgrounds. So we can make multiple cells that have, say, a muscular dystrophy or multiple cell lines that have this, and we have the perfect controls, and we can see really if those drugs work. So there's a real therapeutic aspect of this that's very, very important. But finally, if we can make a change now from the C to an A at this one little page and make the disease, if we have disease cells, and we're really able to do this with high fidelity, we'll be able to take the disease cells and make the change in reverse. And when you make the change in reverse, you can now take the disease cell and make that into a healthy one. And if that's the only problem that person has, and you're able to actually replace those cells back into the person, that's the cellular therapy. And in fact, this is actually what's being done with HIV right now with a certain type of engineering, where one of the receptors for HIV has been removed, and the cells are being put back into people, and the early trials seem to be working. Now, obviously, this isn't going to be the type of change for the masses in the sense that it's going to be, it's an expensive sort of therapy, but it's for the individual, it could be transformative. And it could be transformative not only to say, the cardiac cells that we're thinking about, but liver diseases, blood-forming diseases. Those particular tissues are particularly adept at making, essentially being reconstituted and can be transplanted into a person. So in summary, we've gone from a time where we really couldn't study human disease or couldn't study human genetics to a point where we can actually make cells from patients who have disease and study them and see that, in fact, that we can really model the disease in these cells, as Deepak spoke about. But now we can actually not just observe this, but we can do experiments where we can actually say, now we think that this is the gene which caused it. Now we can make that change, and we can make real models, and then later, I think we can also use this for therapeutic purposes. So thank you very much.
We were the only ones allowed slides, so you get a treat. So my name's Gaia Skibinski. I'm a scientist in Steve Finkbeiner's lab, and we work on several different neurological diseases.
I'm Mike Ondo. I'm a graduate student in Steve's lab.
So we've heard a lot this afternoon about using iPS cell technology for potential therapies and also for generating disease models. And really the take-home message about when we talk about developing a disease model is that we're simply comparing the healthy cells with the disease cells. So this is very analogous to playing spot the difference, which is what you kind of did when you were kids, right? So some of those differences can be really easy to identify, some can be a little bit more tricky, and some of them can be really hard to identify. So if you really want to capitalize on this iPS cell technology, you really need to build good tools that enable you to identify these differences.
So to address this challenge, we developed an automated platform we call robotic microscopy, which we use with disease models such as iPS cells. And so this platform really has three main components. The first is the robotic microscope itself. So we're now up to the third generation, and we're building the fourth generation. This is a fully automated system, which means that you can put your experimental plate on, it'll retrieve and image single cells and return it without human intervention according to the timescales that match the biology. So this can image a single plate in about 15 minutes. So in a single day, it can generate images of hundreds of thousands of neurons. So this brings us to the next component, which is the image analysis. This is crucial to try and automate extraction of the important biological features in our images. No scientist and really no team of scientists can really go through hundreds of thousands of neurons in a day, even for a single feature. And if you ever talk to one of us and ask, what do you want to see in the image, we can just give you a laundry list. And so really, this is working with people who are experts in computer vision to try and extract these relevant features, especially as these features evolve over time. And we're actually quite fortunate already to be working in collaboration with Google on developing the latest pipeline. And then the last, really the foundation of what we do is the statistical models. So this really enables sort of the powerful extraction of the powerful data that we can get from tracking the same cell over time. And really the way to think about it is we basically enroll all of our neurons in a clinical trial where each cell is like a patient. We can ask it questions like, do you smoke? How's your diet? And then we can try and see how that relates to the eventual fate of that cell.
So this is an example of some of the images that we take. And so the way that we track individual cells and the characteristics of those individual cells that you can think of as people is that we use fluorescent proteins. So this is just one image of some brain cells that we're looking at. And each of these green structures is an individual brain cell that we're following. And then the numbers next to it are just the number ID that we use to track the individual cells. And so This is just the image on the first day, but we can actually then image the cells on the next day, the day after that, or for 7 days, for a month, for 2 months. We've actually followed cells for several months. We just get fed up of feeding them, so we stop following them. But this video is looping round. So this is just following a bunch of cells, around 60 brain cells, for 5 days. And so if you keep your eye on 29 and 45, you'll see that 29 starts off well and then dies pretty much immediately, whereas 45 sticks around and then dies at the end. So this is an example of just 60 to 70 cells that we're following, but we have the capacity, as Mike was saying, to follow hundreds and thousands of cells each day. And so there are several key advantages of this system of tracking individual cells, like in a clinical trial. The first is that unlike conventional approaches that just take a single snapshot at one time point in the life of a cell and looks for differences between the healthy and the diseased cells, we're actually taking snapshots of the same individual cell throughout its lifetime, and we're doing that for hundreds and thousands of cells. So our ability to pick up differences between the healthy and the diseased cells is much greater. We have many more opportunities at picking up those differences between the healthy and the diseased cells. So that makes our system far more powerful and sensitive at picking up the differences between the healthy and the diseased cells.
So another advantage is that we can tell if these differences are pushing or pulling a cell towards a particular fate. So again, to go back to the clinical trial analogy, this would be like asking, well, if you started smoking early but you have a healthy diet, how do each of those influence your eventual lifespan? And this sort of understanding is key to understanding disease and the mechanisms that underlying disease. And in particular, if you're trying to develop a therapeutic, you need to understand where and when to target.
Yeah, so in addition to understanding the mechanism of the disease and identifying the differences and what role these differences play in the disease, we're also using this platform in collaboration with many different biotech companies and industry partners to screen for compounds that could potentially identify novel drugs or also new targets for therapeutic strategies for many different neurological diseases. Anyway, so if any of you are interested in coming up to talk to us about this system later, we'll be around at the reception. So feel free to come up and ask us more questions.
Thanks.
Okay, we've given you a necessarily brief set of vignettes about new technologies for life science at Gladstone. And we're now going to move to the second phase of the program. I'm going to invite our panelists to come to the stage, and I'm going to invite any of you to stand up and move around a little bit. Two hours to sit at one time is too long for me, but move around a bit if you like. In the next hour, I want to look back to the way I opened the evening tonight. I commented that there had been a miracle down the road, not too far from here, that I call the Silicon Valley miracle. And I have to tell you, to my own discredit, I did not see that coming. Some of you did and participated in it, but I have to say, somewhat shamefacedly, I did not, and I had a chance. In 1966, I was a college freshman, and I taught myself how to write computer code. My motivation was not noble, it was simple. I needed a summer job. And my always insightful mother called me up and said, IBM is coming to my hometown to open the first computing center, and no one in the state knows how to write code. If you can write code, you can get a summer job. And so while my friends were flipping burgers or washing cars for 75 cents an hour, I got a job with IBM. So I learned how to do it. And I kept it all through college. I earned handsome fees writing programs for computers that were as big as a truck. They were fed only by little cards with punch cards, and the output rolled out on large sheets of paper with little spooled out with pins in the side. Some of you will remember that. Now, I never imagined that someday a device in my pocket would have 1,000,000 times more computer power than the things I was writing for, it just didn't occur to me. But I wasn't all that dumb. I saw a different revolution coming, and that was the revolution in life sciences. The genetic code had been broken that very same year that I learned how to write code for computers. And somehow that caught my imagination more than the future of computing. And that proved out not to be such a bad choice. There have been tremendous discoveries over the decades that I've been fortunate to work in medicine and molecular biology. But I still am struck by the contrast. The revolution I didn't see coming has changed the world in ways that's evident to everyone, and we haven't done that yet. Now, I wasn't alone in this myopia. IBM didn't see it coming either, okay? And they were almost swept away by the waves of innovation that came out of Silicon Valley. They survived and regrouped and were the great company now that they were then. But some radically and almost unimaginable things happened. So I wanted to merge these worlds, to take some of our highly creative innovators from Gladstone and ask some superstars from the Silicon Valley experience to come, and let's have a conversation about this. Why is it that information technology and information sciences penetrated ordinary life and we have not? Is this discrepancy in the very nature of our work? You know, you could say working on human biology is just too damn hard. It's too hard. But that would be a cop-out. I'd like to think that these folks saw some things that are out there that we haven't seen yet in our operating models, in the way we go about our work. There could be, just like for IBM of the 1960s, disruptive changes in how life science is done that are on the horizon. And we want to be those folks. We don't want to have that hit us. We want to be that. And that's, I think, the purpose that we have today. Now, what we're going to do is I'm going to begin in focusing on our guest from Silicon Valley to pose a statement and ask a question. And we'll have some discussion about that, and then we'll pause, and I'll ask one of the other guests to comment. So we're going to begin with Andy Cunningham of Apple fame. And Andy, why don't you tee this up?
Thanks, Sandy. So I'm a marketing person, so I was fortunate enough to be in the right place at the right time and worked very closely with Steve Jobs on the launch of the Macintosh, and got an incredible marketing education from him early on, and continued to work with him for about the next 6 years. So my observations of all of this are going to be very marketing oriented, and I'm not a scientist, although I have amazing admiration for you guys who have done these amazing things, which really are changing the world, despite what you said, Sandy. But I have 3 ideas or 3 notions that I thought about when Sandy asked the question, and each one kind of begets the other, and it sort of bring a little bit of a picture of what it's like to be in the Silicon Valley realm, maybe to you guys. And the first thing is this notion of good enough is good enough in the world of high technology. And this is something that frustrates many people, but often you'll pick up a piece of technology and it doesn't work, or you put your slides up and they don't work, or you turn on your phone and it doesn't work. In the world of technology, we are all okay with this. We're fine with it. You're all okay with it. You spend money to buy these products, they don't work, you're okay with it. And how this is, and the interesting thing about this is this is kind of always the way it's been in Silicon Valley, but Microsoft sort of perfected this about 20 years ago when they first started sending out software that really, really wasn't working very well. But it seemed like it was a mistake at the time, but it has actually turned into an entire philosophy for running small companies, which is now known as the Lean Startup. Has anybody read the book, The Lean Startup, or know about that book? So it's a whole methodology for how you get a startup up and running. And the idea is you do something, you stick it out in the market, you see what sticks, you bring it back, you fix it. You stick it out, bring it back, stick it out, bring it back. This doesn't work in your world, and that's one of the issues that you have. So that issue about good enough being good enough, which it isn't in your world, begets another issue, which is what I'm going to call regulation stifles imagination. So because you can just throw things into the market in the world of technology, and you can't do that here, you guys have to deal with this issue called regulation. Now, I agree with Sandy. You can't use that as an excuse, but you have to realize that it is an imagination-stifling issue. And so you have to work harder to let your imagination go further to try to solve that problem. I was telling Sandy, I met with a company the other day that wants to do some marketing, and they're a biosciences company, and they have found a way to deal with the oxidation in cells in a brand new way. And the challenge that they have in front of them is they're not really a drug and they're not really a nutraceutical. So my question to them coming from Silicon Valley is, well, what is your new category? What's the new category? And they're like, well, we can't have a new category. There's only two. There's nutraceuticals and there's drugs. And my feeling is, well, wow, that's imagination being stifled by the system that's in front of you, which is all about regulation. So you can't let things like that stifle your creativity. And the last idea that I'm just going to lay out relates to another book called Crossing the Chasm. Anybody read Crossing the Chasm? Okay, this issue does not exist in your world, right? You don't have the chasm. You've got something that I call contradicting the chasm, right? Because the chasm is all about launching a product into the industry early on with early adopters and early innovators, and then letting it kind of spread out to the early majority and the late majority, and then when the product cycle is over with, you're hitting the laggards. You guys are always dealing with the majority. You never get to deal with the early innovators, and you never get to deal with the laggards. So you're in this weird situation where there is no product adoption curve for the world that you live in. So you have to deal with this in a very different way, which means you have to figure out how to develop something that is going to hit the majority early on. So you got three big problems, and I'll just end this by saying, how do you teach yourselves to be more brave? How do you challenge the status quo even harder than you already are? How do you take more risks? Be ready to do battle, because it's all about doing battle in the marketplace. And I'll just end with one statement that Steve Jobs told me a long time ago, and I'm sure it's been printed many times, which is this: great artists steal. And so what can you steal from other industries that can make your product a success? So I don't know, what do you guys think about those theories?
Okay, I'll ask, yeah, that was great.
Can I
For
say
a
something about
response,
this?
Leonard Mookie.
Fantastic comments. So thank you so much. I think our world may be much closer to what you just described than you anticipate. So, but I would like to bring up one challenge, for example, that just came up in our recent scientific advisory board meeting, where the board members were incredibly impressed by the bunch of highly unorthodox thinkers that we have assembled. And our main mechanism of spreading the gospel is by the slow and tedious process of putting publications into the literature that more and more now, they are recognized, and the people in the subspecialty fields, they see them, but in the grand scheme of things, they're easily drowned out. And so one has to engage more and more marketing strategies to ensure that the highly innovative ideas actually spread through science, and to make our approaches, like this fabulous microscope that you just heard about, the robotic microscope, the gold standard in the field. So, and for us, oftentimes we want the competition to work on it as well, rather than keep it all inside. But I'd love to hear some thoughts that you have, what one can do to convince others about the validity of one's own unorthodox ideas. So how can you make the world see faster that it's not flat but round? We struggle with this oftentimes, that we think we've got it and we understand it's round, but we're up against a very dogmatic field that needs convincing, and our publications are often not enough.
Yeah, and they're kind of boring too.
What can you teach us to go out there and...
I think marketing matters. It matters in everything. So
Right.
the way you write these papers, can you think about reinventing them in such a way that the regular person would like to read it?
So, many people are starting webinars and are doing, or chat or forums or things like this to just get the buzz going about what they do. And there's sort of a fine line between being, almost obscene in science and actually simply savvy,
Absolutely, yeah.
Okay. I might add, we will open up this conversation to all of you in the audience, but we're going to save that sort of for the second part of the hour. So any other responses to Andy? Okay. Dennis, it's your turn. So Dennis Coleman, please read about him in your program, but of Symantec fame and other accomplishments.
Thank you. It's a pleasure to be here. Young entrepreneurs often come to me and say, Dennis, do I have a business? And I might also add now, old entrepreneurs also come to me and ask the same question, which is interesting because a lot of entrepreneurs, I think, they don't want to retire. It's a question of retire or be relevant, and a lot of them would rather be relevant. So I tell them, I say, well, what sort of project do you have? I say, if you have a software project, estimate all your costs and your time, and then double it. If you have a hardware project, estimate your costs and your times, and multiply by 10. If you have a life science project, estimate your costs and your time, and divide by 0. So the question I have, and I'm going to answer it first for myself here, and get a little backhanded compliment and a backhanded back flip that didn't work. Name an example of a startup you had that took a technology out of a university or a new technology, a little bit brave on the technology side, that was successful, and what was the key reason it was successful? And secondly, take another one that failed, and what was the key reason it failed? Well, the first successful one was Financial Engines, Bill Sharp, Nobel Prize winner, and applied asset allocation to advising retirement programs. And after 14 years, it's now a public company worth a couple of $2 billion. A long struggle. But it was successful because we had a good team, and I think the key was we separated out the technology from the business people. And it was sort of clean to do that here. So Bill and Dr. John Watson, what a great name, they were just always working away happily on the models, and they were far enough ahead that the business people could then get out and solve the problems. And another one, we were using infrared spectroscopy to examine the effect of drugs on protein, the conformation. And the key thing here is we're going to use silicon sample holder etched tiny samples run in parallel. That company today is a piece of history. The investors lost everything. It was a brave effort, but I think one of the reasons it failed is we got so excited about the business and what it could do, we never took a look and said, do we have the real concrete proof that the science works? And the answer is, looking back on it, I can't believe it. We never had that experiment, the reflections, all kinds of funny things were going on with the light, and we never really had that. So let me ask, does anybody else have a case they would like of a successful use of technology or a unsuccessful one with a primary reason? Micah, have you had any? Okay, Micah, have you had any unsuccessful ones?
Well, I was an investor in the company that Dennis just referred to, and it was amazing that the science didn't work in the end. I always felt, Dennis, that the physics the signal wasn't strong enough to support a product, and I think in the end that might have been the problem. I'm not sure how we would have known that up front. Maybe there's some kind of due diligence we could have done.
We would have invested in the equipment and the experiments to find that out, as opposed to investing in how to make it run in parallel and automate it.
Yeah.
I would make the following comment around this point that harkens back to the first part of the program today, and that is drug development up to now has relied upon very imperfect predictors of what might happen when the drug actually goes into real live people, because it's been based on animal models, which we know to be useful but sometimes misleading. And to the extent that human cells were used to pre-screen drugs before they went into humans, these were very weird kinds of human cells, HeLa cells, the famous cells from Henrietta Lacks. Things like that were nowhere near what cells are in our own bodies. But this revolution in IPS technology And cellular reprogramming is now giving us authentic human cells that are the real deal. And they're not only the real deal of humans in general. If we take those cells from you, they're you. So it's very personal. So I think the technology is beginning to give us some of the tools to more closely validate the science than we've ever had before.
So when you build a business, you have to keep, you got your business, you got your funding, and you got your product development, and you're trying to keep them in sync. And if one gets out of sync, you'll fail. And if you don't have the funding, you're going to fail. But if your funding gets ahead of your business, it turns out there's just a lot of paths to failure when that happens. You build the wrong team, you pay them too much, you made the promises to investors, and when you make a promise to an investor and it doesn't come true, what you do is you make a bigger promise to an investor, et cetera, until you fail. Now, when you have life sciences here, you got a fourth element you got to keep in step, and that fourth element is you got to keep your science in step, and that's probably the hardest one. And as I said, that is the one where we failed. Our science simply wasn't in step as we raced ahead with the fundraising and the business and the product, but we never really managed to tame the science.
Okay, thank you.
Comment on that? So, in keeping with the theme of this is what can we in life science steal from the lessons of the technology industry? One thing you brought up resonated with me in that each faculty member at Gladstone and any university is essentially running a small startup with their laboratory. They have to acquire the funding for the science. They've come up with the ideas. They have to direct it. And when you're saying what are the things that would lead to success or failure, when I look at the investigators, scientists who succeed versus fail, it's many of the same qualities. And a lot of it's about leadership, but also the funding issues and getting ahead of your science. I've seen all of those issues. I think there are a lot of parallels that we can learn in the sciences from running a startup. It's essentially the same thing.
Yeah, one other comment from me. I think there's been reluctance for a long time in many circles. This is changing today in a healthy way, I think, to even use business analogies for what scientists do. I think a decade or so ago, you demeaned yourself at the faculty club if the word marketing crossed your lips or return on investment. You put yourself apart from the purity of the intellectual effort. My personal view of that is hogwash. That's a kind of arrogance that we shouldn't tolerate. And as Deepak pointed out, every one of our laboratories is in effect a startup and a small business. We try to nurture them, and like you do to your companies when you're an investor. But there's more similarities in our worlds than I think many people appreciate. Okay, we'll move to the other end of the table. Micah Siegel, you'll again read about in the brochure, has walked the interface between academia and industry and investing probably more than any of us, being a professor at Stanford as well as involved in nurturing quite a number of fledgling companies there. So Micah, your turn.
Sure, probably not more than Dennis, actually, if I look back over the years.
I'm older.
I don't have a prepared statement, but I think reacting to what Dennis said, I do think that there is a sense in which the most successful companies that have been life science related that I've been involved with, and there haven't been that many, all had a sense of a customer early on, and I haven't personally invested in anything that was a pharma compound or anything that's super capital intensive for a variety of reasons. But the ones that were most successful, I'm thinking back, were very early on able to capitalize on some kind of customer outside of the university, and very quickly became customer focused. And the venture dollars, by the way, in most cases followed after the customer dollars. And I think a really good formula. So it's complex. I think that to the extent that life science companies are very capital intensive, you need to have very, very big markets to justify on the other end why you put so much money into them. So you're left with a conundrum if you've got ideas which are evidently commercializable but aren't enormous markets, at least don't appear to be obviously enormous markets at the beginning. What do you do? And I think the answer historically has been that you find customers to fund what you're doing. not in the form of grants or anything like that, but really, if you've got a device that's a fancy microscope that tracks 3,000 cells at once, you ask yourself, well, who wants to buy one of these things? And you sell it to them. And it doesn't matter if you can only make 2, you sell your first one. You keep one, I guess, for your lab, and you sell the other one, and then you make another one. And there's a long history in the, as I look back in investments that I've made that have worked out with that formula for success. So they tend not to be fancy disease targets or drug screening technologies. In fact, interestingly, a number of the ones that start out doing what they think is the sexiest part of the market, which is like screening for new drugs, end up doing one of 2 things. Either they have set very high investor expectations, and go off and realize that they can't make a big enough business doing drug screening alone, in which case they, one by one in the late 90s, became drug companies. And they sort of each announced, well, we've got this super duper platform, we're going to find our own drugs. And I don't know of any one of those, maybe there's one I can think of that succeeded, but that's a pretty big crapshoot. The more successful path I've seen has been the ones that use customer funding early on to grow into the size of the market that they exist in. And if they don't take a lot of outside capital, then, I don't mean to be crass about it, but if the company is finally sold for $50 million or $100 million, but it's only raised $1 million, or even nothing at all for that matter, the founders and let's say the owners do really well. Whereas if they'd raised $40, $50 million along the way, they end up with pretty much nothing. So I did sort of a long-winded ramble, but the formula I've seen is focus on customers early on.
So I'd ask our scientists, who are our customers at Gladstone? I mean, ultimately, it's the patients whose diseases we would prevent or treat. But they are several steps removed from us and disconnected from any economic model that we might apply that principle to. So who are our customers?
Well, I mean, I would say that, there are many things that you can do in industry, fortunately, that we don't have the luxury of doing. So, many pharma companies have walked away from Alzheimer's disease and so on because they consider it too risky. Well, we have the responsibility in academia to fix this problem. If we don't do it, nobody else will. So I think there's a decision we cannot make. We're going to stay in that game until it is solved one way or the other. And the investment for the nation is $200 billion per year, and the failed trials so far have cost $1.2 billion. So it's a relatively small, failure relative to the amount that the nation has to invest into that problem. So there are some math calculations here of, sooner or later it will hit all of us, and there's a huge expenditure on this. But the angle that we have in our math calculations is a little different from what you were just outlining. So I think what drives us oftentimes are more these kinds of calculations besides our very own budget, which of course also has to make sense.
Yeah, Leonard makes a point that I'd really like everyone in this room to understand about Gladstone in the life science medical industry. If we had shareholders or investors to pay off, it would be our moral obligation to make decisions to create profit margins to do that with, right? That's what commercial firms do, quite rightly. But we have purposely chosen to be in the not-for-profit space, and we don't tune our operations to the most lucrative activities. We tune our operations to these long-term societal goals. We're in the business of promoting public good around a solution to neurodegeneration, a solution to heart failure, a cure for HIV. That's what we're in the business to do. And the commercial products to do that with are several steps removed. But nevertheless, I don't think we should shy away from this business terminology. We have customers that we have to satisfy to gain the means to work in that mode. So any other comments about who are our customers?
I think in this case, because science works at a global level and in mass, our customers, I think, are our scientific peers.
Exactly. That's the first.
Because we have to, as you were saying, convince them of a path forward that they would then, we create a momentum for, as maybe a leader in that area, but we create the momentum so that the field moves closer to our ultimate customer, which is the patient, but it's so far removed. But our first customers, I think, are our peers, our scientific peers.
Our peers. And this has a very important business principle, too. Roughly half of the funding for Gladstone comes from government grants that are competitively reviewed by our peers. So to the extent that our peers think our work is meritorious, that drives the largest revenue stream for us. So that's definitely a customer. Katie?
Yeah, I was going to say something along similar lines. I think it's important to ask who the customers are, and I agree that they're our peers. And then the question is, what is our product? And I think that's also an important distinction that we're kind of dancing around here, which is we're in the business of generating knowledge, not iPhones. And so there's some fundamental differences there. And some of that knowledge can go across the barrier onto the industry side and lead to products, and that's happened really effectively with a lot of my colleagues in the computational and statistical realm are now working in companies and using academic knowledge, and it can come the other way where we harness technologies to do our work. But the business model is, while the analogies work and I'm all for using the business terminology, the thing we're doing, the enterprise, is kind of different, I think.
It's quite different. Our product is knowledge, but increasingly so is industry. Knowledge-based industry is a whole sector now. Okay, we have Mike Frumpkin with us from Google. Mike, your turn to tee up a line of discussion here.
So when I look at, how do you compare and contrast kind of what's going on in the technology industry and in your world, for me, in some ways, it comes down to people and what those people are enabled to do. All of the progress that we see pretty much in both of these industries, in the end, comes down to individual people creating interesting breakthroughs. And you can talk about how these different people are incented differently in the two industries and so forth, but in some ways it's more about the tools that they have and how efficient can they be. Even in the best structured organizations, people, you build on each other's knowledge, but there's communication costs. Anything that involves a lot of people in some ways is less efficient, and a lot of progress is really made by single individuals. In Silicon Valley, over the last 25 years, longer actually, but it's accelerating, the amount of interesting contribution a single person can make has increased tremendously. If I look at writing software in the late 80s or early 90s and writing software now, the amount of work that's required to produce a finished product that someone wants, required in the late 80s, is orders of magnitude more.
About the 60s when I was doing that.
And punch cards made it even harder. So I think if I look at the life sciences, how much of your day for a research scientist is spent really thinking hard about the problem, and how much of it is spent on tools, on overhead, on those sorts of things. And in the end, if you realize that it's the people that make the breakthroughs, as you build better tools, and those individuals get much more efficient, I think you have very much a multiplicative effect on what the industry can produce. And the good news is, if I look, at the tools that you guys are building, I think you're heading that direction. So I think the analogy is potentially an interesting one, just time shifted a little bit, because as you make the individual researchers, truly utilize what is their most valuable contribution, which is their brain power and how they think about these things, and more of their time goes to that, you're going to greatly increase the rate of progress.
Well, I'd like to carry that theme on in the following sense. Most of the nonprofit life science world are in universities, right? Universities are quite proudly medieval in their structure. They are. They even use medieval terminology like deans. So the university and its departments, the roles of faculty members, are among the longest lived structures in all of human endeavor. This is good because universities last a long time, right? They last longer than most countries. It's true, you can count it up. I heard a talk about that last year. Now, at Gladstone, we've not a university. We've moved aside, and we'd like to think we've left behind some of those more medieval type structures. But I have to say, we are still running our labs the way labs were fundamentally run in the 1950s. The basic model of doing life science research has not changed, the organizational structure. So, you know, what are we about to be overcome by somebody who figures out a way to accomplish what you've said?
So I think we should be stressing more the role of marketing in what you do. Now, if you cure Alzheimer's, of course, you probably don't need to market that.
Right.
But I was talking to Regis McKenna, who's an icon. I'm sure you know him well in marketing. And he was at a meeting where they were celebrating the technologists. And he was saying, well, they don't celebrate the great marketing people. So he got up and he said, you know, the product sells itself. But that's just not true. Without Steve Jobs and his presenting it and his shows and everything here, the thing never would have been anywhere near what it has. But it seems that you've got so many. The way you solve hard problems is just a lot of different steps, and you're involved in 20, 30-year steps, of which you've obviously taken some very big ones. But each one of those things has to be marketed and explained, and there's just a lot to learn by just seeing how successful people have learned how to market their products. And it often takes a company, you know, even Google, years before they come up with just the simple statement that you can write on the back of a business card. It starts out as something long and pointers and links and better, and eventually gets best damn search engine and get done with it. And I can think of an investment that I made. I like to tell good investment stories in front of Micah here because he knows about all of my bad ones. And there are lots more bad ones than good ones. 3 years ago, I went to a friend, and he's a young guy, and what are you doing? And he explained it to me, and I said, oh. I went back, and I saw him 3 months later, explained what he was doing. I said, I want to invest in that today. And by the way, what's that thing that you were doing 3 months ago? And he says, Dennis, it's exactly the same thing. We've just learned how to explain it. And he just closed $40 million at a $345 million valuation on very small sales.
So there is another, and I know what investment you're talking about, and thank you for letting me invest in that in a little tiny bit. There's a version of marketing, it's a little off topic, but it's maybe relevant. In the academic world, putting aside the commercial success or lack of success, I've seen interesting kinds of marketing. So you're right, you mentioned at the beginning, I think, Sandy, that I think you mentioned that scientists can be very wary of marketing, as if the science should speak for itself. And I was a scientist before, so I understand that feeling. That said, I've noticed in the field of optogenetics, the guys at Stanford have been very shrewd about what I would call marketing, and I mean that in the most positive sense. What optogenetics had was a really revolutionary technology for stimulating brain cells, but it was hard to use. And I think that there, like a lot of these things just beginning, there's a lot of know-how around getting it to work in your lab. And there's a million different applications for the darn thing. So what the guys at Stanford did, they didn't call it marketing, but what they did is they started running seminars for how to use it. They have like resident fellows that come and spend 3 or 4 weeks, I think, at Stanford. They basically made it easy for the customer, in their case other academics, to use the product, which was a new technique. And it's you know, maybe it would have taken off like wildfire without it, but it certainly helped. There's probably a version of that in most any field, whether it's a new microscope or embryonic, some stem cells. There's a way of getting it adopted by your community that's not, it's a kind of marketing, really, in the end.
Yeah.
It was very effective. I totally agree with you. I think it's a great example. Good marketing.
So Andy, and then Mike.
I just wanted to add that I did a project with UCSF about probably 5 or 6 years ago, and it was a marketing project, and we were brought in actually by the, what is that board of higher-ups that run the whole thing?
Regents?
Yeah, the regents, exactly. And it was a marketing project. But when I first started working with them, it was so clear there was no way I could even whisper the word marketing in among the doctors and the researchers. So we ended up calling it a fundraising, which was okay. They were all right with raising money. So, but you know what that was? That was positioning. We repositioned our project for the audience that we were dealing with. And my point here is positioning is what it's all about. And that is what I learned from Regis. I worked for Regis when I first came to Silicon Valley, and it's all about positioning.
So, being an engineer, I'll basically take a slightly opposing view on the marketing thing. I agree that marketing is a very important thing and that you have to be able to explain your products and explain what you're doing to other people. But first, you actually have to have something to market. And I think that's really the thing to concentrate on, and what are the things that can get you there. It does feed back on itself. If you can market things better, you create more success and more incentive for people to do follow-up work and so forth. So I'm not saying it's not important, but I think the really interesting thing is, how do you accelerate the rate of discovery and development? And the marketing comes after.
Can I follow up on that? I think one of the more medieval, to continue the analogy, things that we do in academia is how we communicate or market our ideas, which is to write these papers in journals that take a year or two after the discovery before they come out. They may not come out, they come out in some mutated form. But that actually is changing. I'm sure most of you are aware with the open access publishing movement, and more recently a lot of scientists are just discussing online the work that they're doing in their labs through blogs and other forums. So I think that's kind of like the lean startup idea, that it doesn't have to be perfect before you throw it out. The papers are perfect, or like we at least hope they are. By the time they get through that whole review process and they get published, they should be more like a finished a company,
Right.
a well-formed company. But I think this open communication in science is new and it's just getting going, but I think it's going to be more like the lean startup. And I personally think it's incredibly important.
Exciting.
Dennis?
So remember, marketing isn't just explaining. Marketing is putting your product in front of the customer and then telling the engineers what features they have to implement. Not because you want them implemented, but because the customer wants them implemented. And that would go for your microscope. Go around and find out, what do they want in the labs and what will they pay for.
Yeah, to carry on this customer, who's your customer theme for a moment, it's quite clear we have multiple customers and they speak different languages. Our dominant customer is rightly what Deepak mentioned. It's our scientific peers. And I don't want to change that. I want to serve that customer. On the other hand, we have two other very important kind of customers without whom we can't do our business. One would be the commercial groups with whom we interact. Our ideas will never reach a patient unless some commercial firm picks it up and runs with it. And in optimizing our product for the academic audience, we often de-optimize it for the commercial audience. And getting that balance right is tough for us. We're still learning how to do that. And then there's a third audience I have to acknowledge, and that's you. That's the people in the room here, a third set of customers. One of our customers has to be the people who don't work here, but who believe in what we do and support us. And without that, we can't carry on. So we have to speak your language too. And this notion of throwing things out and getting them back from you, that I want to encourage very much. So please do that for us in acknowledging this principle.
Sandy, when I've seen a lot of descriptions of companies coming out of universities over the years, I would say that the problem with most of them is they don't know what business they're in, and they don't know who their customer is. And when you, not to pick on you, but you're standing up, so I'll pick on you.
Please.
When you say you have 3 customers, I hear as an investor or as somebody who would join a company like that, I hear that it's unfocused. And I don't mean that as a criticism. I mean that it's just, in my experience, better to have one customer and service that customer ridiculously well, and the rest will take care of itself. So, for example, if it's a granting agency, then you need to raise money from grants, and that's your customer. What activities do you do to service that customer? If it's patients, well, patients aren't paying you directly, so I think they kind of get ruled out. Your peers don't pay you, so they're ruled out. Unless they're doing a service. If you're building a microscope, they might pay you.
They're reviewing the grants, though.
Fair enough. So I've found it very successful over the years to just follow the money, and whoever's writing the check is your customer.
That's a simple principle. So one more theme to pick up, and Katie brought this to my attention in the preparation for this session. She posed the premise that, is there a very different cultural distinction we draw the line of distinction between academia and industry differently in the engineering, information sciences, technology world versus life science. It was quite clear in the past we did. When I grew up, the people who worked in pharma companies, there weren't any biotech yet, were just completely different from the people who worked in universities. That's changed a lot now. There's a lot more movement back and forth, but we still have a fairly bright line distinction. How does that work in your world? Is it quite different? I have a sense it might be.
What's the question? I'm sorry.
Is the distinction between what's academic and what's industrial a bright line or a blurred line in terms of people and the way they define themselves and the way business is conducted?
I actually do have a quick comment about that. So you might be familiar with IDEO and the design school at Stanford. I think that those guys have done a phenomenal job of bringing the two sides together in a way that serves the commercial market and serves the academic market. It's just brilliant, I think, what they've done.
Well, I think this goes to, I taught for 6 years. And now when I go back and I see my friends that have taught for 36 years, we really live in very different worlds where our motivations are different. It's just quite different. And when you want to spin a company out of a university, I think it's really important that you get all the capability out of that university, because a person in the university simply has other things that they're doing, other criteria they're evaluated by, and their life is quite different. And when you go to them and something doesn't work at the last minute and you need to get their attention, it simply isn't there.
Okay. All right. I want to save time to let the audience come into play here. So please step forward.
You know, the 360 view of customers is starting to permeate the world of enterprise marketing now, which I think is a great thing, which is there's more than one customer that you're dealing with. And so we're starting to actually learn about that, and you guys have done that for a long time. And I think, well, again, it's really about positioning. It's about what is the ideal position. If you get the ideal positioning for a product or a drug or a treatment or whatever down at the highest level, parsing that out to the various customers is actually easy to do. What most people don't do well is they don't understand what that high-level positioning is, and so they're working from the bottom up, and it's very difficult to do it. But you got to think about the 360-degree customer. It's everybody. So
yeah, I just stay away from companies with complicated customer buying patterns. It's too hard. You know, I'm serious. It's already so hard. You know, it's like so hard to get something out of university when it's. But that, you're talking about drugs. So if I don't mean to beat up the microscope. I just heard about it, right? So if you had a microscope and you wanted to sell it, there might be the distinction, as you know, between the person who decides to buy it, like the PI, and the person who'd be using it, like the graduate student in the lab. And some of those dynamics are interesting, but they're not that hard to get around. So I think the analogy is interesting between the two.
Well, I think this goes down to follow the money here, if I could be so crude about it. In the healthcare industry, people pay their insurance and then expect everything to be free. They expect their disease to be their doctor's problem. And nobody really knows how much it costs until somebody gets a bill, and then that person is very unhappy. And you just have to get all the reimbursement, and you have to get that down before you're going to find investors like Mike who will invest in you.
Pitch?
Well, I'll make a comment about it. I think you're doing marketing the minute you choose what disease you're going to go after. That's a marketing decision. And you may choose the right one and you may choose the wrong one. I mean, marketing decisions are made from the very get-go. You just don't recognize them as marketing decisions, but they are.
Yeah, this is where I think we could be at an inflection point in life sciences in the next decade, because we're on the threshold, as you heard earlier, of having the analytical tools and the experimental systems in hand to do that. I estimate we know 1% of what we need to know to actually do rational drug design most of the time, or have things that have some confidence that they're actually going to work. But much of that remainder, I think, looks to be soon within our grasp.
Well, and I think this again goes to the maturity of the tools in the two industries. If you have a great idea at the life sciences, it's very likely that it's going to take you years, if not longer, to actually try and see if it works. Whereas as the tools increase with the things you're doing and the comparison to, the software industry, you could probably try something fairly simple much faster, and so you iterate a lot more. And I think that actually opens up basically a different way for people to look at innovation. If you can try a bunch of things, you're going to take a lot more risk. If in your entire career you're going to get to try 4 things, you're much, much less likely
But this
to take
comes
risk.
back to our operating model. Remember, we're a collection of small entrepreneurs, and if they go down and fail for a limited period of time, they're out of business. Now, we'll prop them up for a while, but there's quite a short string on that, and which is why I think we should be exploring some new models of how we organize the way we do science. And as some of you know, we have some ideas about how to do that.
Well,
Yes, here, sir.
Certainly, I think that from my interactions with industry, I've been very impressed how in industry no-go decisions are made very readily and oftentimes, in my opinion, too quickly. Whereas the academic side errs on the other end, where I think we are very bad at making no-go decisions, and in fact our whole academic assessment process disfavors admittance of failure. And so I think we can learn from each other in great ways. I think sometimes no-go decisions in industry are made because there isn't enough time to really drill down deep, or people don't stay in one place long enough to develop the kind of depth you have to develop, a love affair with one molecule for 30 years, for instance, to really clinch it, So I think that, in my opinion, the best way forward is probably in the middle. I think that at the moment, no-go decisions don't happen readily enough in academia, and we should probably encourage it a little more. And I think I'm very concerned about the enormous fluidity in industry, where the time frames now are truncated more and more, where you have to be successful in shorter and shorter time frames when the remaining problems are actually much larger than they used to be, requiring much more investment of time and effort that people can't afford to do. So I think here, I hope that solutions will come at the interface between academic and industry relationships, of which we've seen a very gratifying increase in recent years.
Yes, please.
I think ethics are important in anything you do. I agree you have challenges, but it's very important in business also to be ethical, I think. But that is a challenge. I love this open publishing thing. I think that's a fabulous way to deal with some of these issues that you are facing. So I don't know, it's a cool development.
So I think you have to take a gulp here. If you have a database, somebody's using it to run a mail order, it doesn't work, so what? You have the simplest little thing where someone is describing a condition to their doctor. And if something goes wrong, it goes to the wrong person. There's some data that isn't there, then somebody can be hurt. And if you think long enough about it, you're never going to do anything.
Yeah, I launched an airplane once, though, and I got to tell you, there's safety issues. The same stuff that you deal with is true with something like an airplane. I don't think Tesla had quite the same challenges, but anytime you're launching a vehicle into the marketplace that does something that carries people around in it, you're dealing with very, very heavy-duty ethical issues.
Okay, I want to thank our panel. This was just what I hoped for. Thank you. And for those who have the stamina, there's one more part to the Gladstone experience this evening. We'll break up now into 3 interest groups. Some of you have pre-identified yourself as wanting to hear most about heart failure and congenital heart disease. You should head to classrooms A and B in that direction. If you want to hear about type 1 diabetes, there'll be accomplished Gladstone folks to talk to you about that in classrooms C and D. And those who want to hear about neurological disease and our attack on that should stay here. And we'll spend about a half an hour at that next session, and then we'll have a drink at the reception. Thank you.
It's my great pleasure to open this session on neurological diseases. What's that? All right, thank you very much. So just briefly, I think you have the bio sketches in here and the names. And so this is Alexandra Nelson and Ken Nakamura and Keith Vossel. And I'm Leonard Mucki. I direct the Gladstone Institute of Neurological Disease. And we're all physician scientists, so we see patients. And some of us, I really want to applaud Alexandra, who dragged herself here despite an accident to be here and answer questions. So I hope this will work out. And so this shows you the kind of dedication that we have represented here up front. But so what I would encourage you to do, if you have questions about anything relating to the brain or spinal cord or peripheral nervous system, bring it on. And ask us, and there are some different areas of subspecialization represented here, but we're very happy to answer as best as we can. Yes?
How far away are you from actually
So the question is, how far are we from using stem cells to actually cure Parkinson's disease? Should I try this, or let me just answer this? So it depends a little bit on how broadly you define use. So there are already models that have been established. So for example, Steve Finkbeiner here has established an iPS cell model of Parkinson's disease. So he's used iPS cells from a Parkinson's patient and turned them into brain cells, and is now using those models to investigate how the disease works and what drugs might be most effective in blocking the detrimental processes that play out in these cells. So that is clearly a use of iPS cells applied toward curing Parkinson's disease. Now, if you're thinking of differentiating these cells into the kind of nerve cells that are lost in Parkinson's disease and transplanting them into the brain of patients, I think that people are actually getting reasonably close to doing this. There are some very exciting studies going on with, not iPS cell-derived stem cells, but embryo-derived cells and fetal grafts. And maybe one of you two can comment on this, or Alexandra also.
I'll comment on this. I think I treat many Parkinson's patients, and this is probably the most common question that I receive. And, we know that if we stick a tube into someone's stomach and give them levodopa continuously, that they will perform better. And so if we have a continuous delivery of dopamine to the brain, things go better on average. So in that sense, we may not be too far away from being able to use stem cells to make a continuous pump within the brain. But at the same time, that's going to be a long ways from a cure. And there's really 2 reasons for that. The first is that normally you have dopamine neurons that are in one part of your brain, in your brainstem, and they come a long ways forward, and they make connections. And that we have no way of remaking that connection, remaking that pathway. So the stem cells right now are just going into the target area. They're not starting at the right place. So it's only a crude reconstruction of the pathway. That's one reason. And the other is that we know that Parkinson's disease now is much broader than just loss of dopamine neurons. And the dopamine neurons, when they're lost, they cause the movement problem. It's a very important part of the disease. But especially as the disease goes on, it becomes much broader in many people. Many other systems are affected. And we have nothing is on the horizon in terms of being able to correct that aspect. So I'd say, in one sense, we're probably not too far away from being able to impact this one important part of the disease, but we're really a long, long ways. I mean, there's nothing on the horizon right now for using stem cells to fix the disease in its totality.
And another important caveat about the implantation of new cells into a diseased brain is that you put them in sometimes a very toxic environment that caused the disease in the first place. So I think it may sometimes be necessary to either make the transplants immune against those factors by genetic engineering, and we're working on this here actually to see if we can genetically engineer cells while they're still in the dish and then transplant them. This works in mice very effectively in models of Alzheimer's disease, as some of our investigators have recently discovered. But there may also be the need to combine transplants with drug treatment that actually gets at the root causes, so the accumulation of all the poisonous proteins that ultimately are going to disable the transplants just as they did the original host brain. So those are things that, have to be tweaked. But I would say, it is something that we can at least seriously talk about at this stage. There are always obstacles and challenges, but in experimental models, this is very much going on. And so, I wouldn't want to put a certain year on this, when this will happen, but I think, there are clearly such efforts underway.
I'll say also, I've come to gain an appreciation for the potential of stem cell therapy. I initially, starting out in science after neurology residency, have been told by others that using stem cells is kind of like for Alzheimer's disease would be analogous to pouring silicon on a computer that's melted down, because the brain is so intricately wired and so complex. It seems almost too futuristic to replace a lot of those complex networks. But I've come to understand that with certain cell types, one can actually take cells that already have the programming to migrate where they need to go. And we've learned how to do this with cells from this part of the brain called the MGE, medial ganglionic nucleus, so basically these cells are inhibitory cells that can help quieten activity throughout the brain. And in epilepsy, a lot of times these types of cells are actually not performing properly. They're not inhibiting the overall network activity like they should be, and this can ultimately lead to excess excitability and seizures. And these MGE cells, we found that if we take them at the right time in embryonic mice, can actually migrate to areas of the brain throughout the cortex and quieten activity in an epilepsy model, and also in Alzheimer's models where epilepsy seems to be a component of that.
Yes.
So neurodegenerative diseases seem highly correlated with aging. Working research on that impacting your work in one direction or the other, insights from there applying to what you're doing?
Yeah, so, as it so happens, we will publish a paper in Cell Reports on May 8th, where we are studying an anti-aging factor called Klotho. This is a very poetic name. The protein was named after the Greek goddess that spins the thread of life. And one of her sisters measures its length, and a third sister cuts it off. And that's what, is said to determine our fate. And so Klotho, this protein, is known to increase longevity, but we discovered that it also enhances cognition at all life stages that we've examined so far. So it's very interesting. And so one can, in fact, enhance cognition in young mice and old mice, and there happens to be a genetic variant of this Klotho factor that about a fifth of you in the audience have, that increases your mental capacity somewhat, but significantly so. And we found this in 3 different human populations in Los Angeles, here in San Francisco, and at Rush University in the Midwest. And we studied over 700 patients, and not patients, but people. And whoever had this genetic variant that cranked up this clothoprotein, they did better on a great variety of cognitive tests. So the interface between aging and disease is really fascinating for us. And why do some people age more successfully or gracefully than others? And what can we learn from those? So, for example, Bob Maley, who's back there, and Yadong Huang here, are very interested in the question why some people who inherit 2 ApoE4 genes, so 2 of the bad ApoE variants that increase your risk of Alzheimer's, escape and manage to be 88 or 100 and still have proper cognition. So they are studying now the whole genome in those patients via iPS cell models also to try to understand how did they escape, right? Because many people, or most people with ApoE4 don't. So I think the interface between aging as the major risk factor for all these neurodegenerative diseases and their exploitation for therapeutic purposes is incredibly fascinating. And we pursue that very actively. Yes?
So if I could use my favorite steroid analogy here. That is, if I took steroids all my life, I probably would be a wreck. On the other hand, given the finite horizon here, maybe I should take steroids to get a better and easier gym workout. Now, if you take something like Claudio, if it increases these, if one took it for a lifetime, would one sort of use something up?
So it doesn't look like that. So, sorry, the question is if sort of a boost is well placed and well tolerated for short periods of time, but if you are on something for long periods of time, maybe it could be detrimental. And so several things to say about this particular clothoprotein in this regard. Clotho decreases with aging, unfortunately. And so we think that maybe part of the aging-associated cognitive decline could be due to failing clotho levels. And as you know, that when women hit menopause, there is oftentimes some benefit to hormone replacement, or there are certain things that, can be replaced that, work well. If our own body doesn't regulate our blood pressure properly, we tweak it down a little bit and so on. And statins, many people certainly take for many, many years with great benefit. So if it was possible to find a drug, and we're going to work on this very adamantly, to elevate clotho levels, it seems very safe in the fifth of the population that has higher levels probably throughout their lifetime. And so if we could just crank it up a notch, one might get some additional cognitive reserve out of this, or even boost cognition beyond the natural level that one would have. So we're interested in pursuing this as a cognition enhancer.
What is the drug that the kids take to do better on the SAT exams?
God only knows. I mean, there may be a variety. So
Talking about stimulants?
Yeah,
probably stimulants.
Those do
right.
have negative long-term consequences.
So what do the kids take to do better on SAT exams was the question. And so there are, a lot of them are stimulants, but Alexandra, do you have?
I think you're probably referring to, there's been a lot of news reports about teenagers getting prescriptions for stimulants that are prescribed fairly liberally for attention deficit disorder, and then other kids get a hold of the same prescription. And so it's quite widespread, I think, among teenagers, especially at competitive prep schools and that sort of thing, to take these before tests or leading up to tests. But stimulants like amphetamine or Ritalin, which is another common stimulant used in ADHD, do have negative long-term consequences, both physiologically and probably in the brain as well.
Yeah, and I should say that we have no idea what clotho would do in the long run, but everything, I mean, it increases the lifespan. It diminishes the risk of stroke, and it seems to enhance cognition. So far, check. We like it, and I think it may be maybe a good thing. I'm sorry. So there, there is a genetic test. And so how do you test for this particular clotho variant? So it is a, that as I said, about 4/5 of us, the majority of people have what we would call the normal or wild type sequence, and about 20% of the population has some changes in the sequence of this gene that results in higher blood levels of this protein. And that can be simply done through gene sequencing. One could find out if a person has it or doesn't have it. And another question is whether, it's already maxed out even in those people who have this variant. It may be possible to increase it even further. And these are all things that we need to figure out, but we're very eager to do that. I mean, among many other things that we're currently pursuing. I mean, I want to mention and maybe advertise a little bit a trial that Keith is directing in collaboration with the Memory and Aging Center. Can you just say a few words about it? Because it is sort of a great example of mouse to men or mouse to human work that emerged from my lab here and has now actually made it into the clinic. And he's just launching this clinical trial now.
Sure, I'd love to tell you a little bit about this. This really dates back about 10 years ago when Dr. Mookie and his group discovered seizures in the Alzheimer's model called the APP mouse, or that stands for human amyloid precursor protein. So this is basically a transgenic mouse or a mutant mouse that simulates some of the aspects of Alzheimer's disease, essentially by taking advantage of what we know about the genetics in genetic Alzheimer's disease, which is rare. But we know it's a good model for the disease. So they discovered that these mice actually have sub-behavioral kind of subtle epileptic activity going on in the brain, and occasionally they actually have full-blown seizures. And actually, a lot of labs have been studying these mice, but didn't realize that these mice were having seizures because they were so subtle. And they finally discovered them through electroencephalography, which is another a fancy word for EEG, brainwave study, where one can see the signature features of seizures, including these little spike waves throughout the brain. Entering Dr. Mookie's lab, I had seen numerous patients with Alzheimer's disease in the hospital and had not realized that seizures are actually quite common in the disease. And in my time here, I've basically was curious about how common this is in the human condition. So I began an observational study where we were enrolling people with Alzheimer's disease and related conditions and actually hooking them up to long-term EEG, long-term brainwave study, and an additional cutting-edge measure called magnetoencephalography, which complements the EEG by recording brainwaves through the magnetic signals that they generate. And through this study, we actually did discover that there are quite a few patients with Alzheimer's disease, even who have never had seizures, who are showing this signature of epileptic activity on their EEG or magnetoencephalography. So based on that study and some additional evidence from Dr. Mookie's lab, we've basically been very interested in screening anti-epileptic drugs on the mice because we have these at our disposal. They're already FDA approved. We screened all of the FDA approved medications in the animal models for Alzheimer's disease and found that actually one of them worked really well at suppressing seizures. And not only did it suppress seizures, but it actually improved their learning and memory. And it not only did that, but actually had some disease-modifying effects in these mice. There are some changes in the brain that occur in response to the seizures that this medication actually reversed. And it kept the brain from having to defend itself against these seizures. And by doing so, the brain became more plastic and able to learn and remember. So basically what we've done now is we're ready to launch a clinical trial for this drug. It's called levetiracetam, very well tolerated. We find that actually very low doses work quite well in the mice and in humans in preliminary studies. So we're excited to get this study launched also in collaboration with UCSF.
So this is an off-label, it's a repurposing trial, a phase 2A trial for levetiracetam, which is marketed under the trade name of Keppra. And it's a very interesting sort of later generation anti-epileptic drug that, as Keith said, particularly at low doses, has a very nice side effect profile. So we think it will be good. And he has a 12-week trial where people will go on the drug for 4 weeks or on a placebo. Then they'll be taken off both types of drugs for 4 weeks, and then they will cross over, and the ones who got the placebo first will then get the drug, and vice versa. And so then throughout this time, they'll be studied very carefully with these measures of brainwave recordings, but also cognitive tests and those sorts of things. And so if you know anybody in the early stage of Alzheimer's disease, which is where we are focusing on, who might be appropriate or interested, they should definitely contact Keith to see if there's anything we can do for them in this work that directly emerged from the research we're doing. So this is sort of the fast track route, whereas in Clotho, this is the long of it for sure, where we have an interesting insight and discovery that we will then slowly want to leverage into drug development efforts. But here we've got a drug that nobody would have ever thought of applying to this condition, but a lot of our research would suggest that that could be done fairly quickly. So if it is positive, that would be very exciting, and we would then scale up the trial in a big way, probably. Yes?
What percentage of Alzheimer's suffers from, whether they know it or not?
I think that is still an open question. So the question is how common are seizures in Alzheimer's disease? And the literature would say between 10 and 20% of patients with Alzheimer's will develop an unprovoked seizure at some point. But that's what we're looking at. And so with the fancy tests that we've been able to do with magnetoencephalography and EEG, so basically with younger onset cases, which we know are at more risk for seizures, we're seeing it in almost half of the subjects.
So are you looking for subjects who are suffering from seizures, or any subjects?
Well, both actually. We would love, so we're basically screening ahead of time for the signature epileptic activity, which is a sign of risk for seizures, and we pick it up we're more likely to pick it up in somebody who's had a seizure, but we could also pick it up in people who've not had an obvious seizure.
Right, so but the trial will focus on people who do have some evidence for this abnormal network activity on the fancy tests that Keith is using in the clinic, because we think those are the cases that have the highest chance of benefiting from this particular intervention. Now, one should say that, this method that we use in all the methods that we have at our disposal are not that great at picking up this kind of abnormal network activity deep down in the brain where most of the Alzheimer's disease starts first. So we think that we're really just looking at the tip of the iceberg and suspect that this kind of activity may exist in an even greater proportion. But it's an interesting way of, kind of precision medicine or personalized medicine, which I think is the way to go for in many respects in the future, where we're trying to identify a very well characterized patient population with a unique aspect and really tailor making a treatment specifically for that group. A lot of problems I think have emerged in Alzheimer trials from lumping very heterogeneous patient groups together in these very large trials. And in many cases, it's now obvious that many of the cases that were included have dementia, but not of the Alzheimer type. Up to 30% of people that were enrolled in very large trials probably didn't have Alzheimer's disease. So the negative results of some of the trials that have been conducted may be as much a problem of the patient population that was studied as of the drug that was used in those trials. Yes?
I'm not sure your institute is doing work in this space, but could you give an assessment of where we are and where we could go in the use of stem cell therapy in spinal cord injury?
Right. So in our institute, Katerina Akasoglu works on spinal cord injury. She's very interested in the contribution that inflammation makes to spinal cord injury, and has some very interesting drugs she's developing in collaboration with Lundbeck that might be important in preventing the problem that results after spinal cord injury. but we have not focused on directly using stem cells for spinal cord injury as a treatment as the people, for example, at Cedars-Sinai are doing in LA. But let me just say one, yeah, no, I think that's all I can say to that.
Let me comment a little bit on this.
Can you speak up really loudly?
Yeah, the question about stem cell therapy potential. Spinal cord injury, right? You know, we showed in this institute we can program directly fibroblasts into stem cells that can generate the neurons in culture by using just single backside tube. Very, very recently, it was actually 2-3 days ago, there was a new paper came out focused on spinal cord injury using that same protocol, but translate to in vivo in mouse model, not in human studies, but they use the same factor plus some other stimulators. And then they can also convert those astrocytes in the local injury area into neurons. What we saw was actually you can see significant recovery, better recovery compared to other control mice with spinal cord injury without this direct reprogramming. So based on that mouse study, I'll say there is some at least indication if there's a proper way to really directly reprogram these on-site astrocytes, because whenever these spinal cord injury occur, many of those local astrocytes proliferate. We generate a lot more astrocytes there. So this new protocol actually tried to reprogram these local proliferating astrocytes into neurons. Seems encouraging based on this.
Yeah, so those are things that Deepak has pioneered in the cardiac area and Yadong has pioneered in the neuro area of circumventing the need of making iPS cell-derived cells and transplanting them, but to give factors that maybe on site, wherever the problem lies, could convert precursor cells into the neurons that were lost by the injury. And that, to me, seems a very attractive and exciting way to go.
I wanted to just briefly comment on some of the interface and some of the interesting ethical issues that I think we will face more and more. You know, because aging, for instance, is the biggest risk factor for many of the diseases we study, and some of the factors that we will find and are finding may impact normal function as well as fixing the disease. And I think, you know, it's very clear everyone agrees that we should do everything we can to restore the normal function when it's compromised in a disease. And I think more and more we will have the opportunity to potentially increase the function even beyond normal, and these will lead to interesting considerations that will really require a discourse with the people and the people, the patients, and the other people in our society.
Yes. So we do not, it's a very complicated condition. And, you know, it is, yes, it really is. And I think all four of us see a lot of this and have seen a lot of this. And we are not currently working on this condition. And I see it in many, at least as far as I know. And, you know, I see it very much as so complex, and I don't see a very good entry point, you know, for decisive investigations. Very much the way I looked at Alzheimer's disease in the 80s. In the 80s, I was working on virological diseases, including AIDS, dementia, and those sorts of things. And I didn't want to work on Alzheimer's disease because I didn't know how to tackle it. And then genetics broke open fantastic entry points, because it was very clear that there are certain genes that can cause the disease in everybody who has that gene. And that was absolutely a game changer. So ApoE4 emerged, APP you heard about emerge, and so on. And we're still missing this in fibromyalgia. And I'm not saying it necessarily has to be from genetics, but it would be great to have a really good entry point for investigation, and unfortunately that still doesn't exist in this particular disease. One more question and then we go and have some wine. Yes, absolutely. So Lou Gehrig's disease, or amyotrophic lateral sclerosis, is something that we're working on very intensely. So Steve Finkbeiner, who is applying the full force of his robotic microscope to very specifically this condition, and has made some interesting iPS cell-derived models also for ALS, and is looking at a particular factor called TDP43, which is a protein that becomes problematic in many cases with ALS. And so we have worked on this also in my lab a little bit, but I think there are some very exciting developments on that front.
Yeah, there's a new gene that was identified just a couple of years ago, partly with neurologists here at UCSF and Gladstone, that's responsible for a great deal of ALS, as well as another related disease, frontotemporal dementia, which is also studied heavily here at the Gladstone. And I think that making that connection genetically is really blowing open a lot of opportunities for understanding ALS, because before we haven't really had that handle that Leonard was mentioning really helps us to dive in with understanding these diseases.
Well, thank you so much for the interesting questions, and we'll be at the reception.